The overall goal of this technique is to prepare enriched sex-specific hippocampal astrocyte cultures from newborn mice as a tool to study the sex-specific roles of astrocytes following oxygen and glucose deprivation and reoxygenation, in order to mimic in vivo hypoxic ischemia in a cell culture environment.
This method can help answer key mechanistic and translational questions pertaining to the sex-specific role of hippocampal astrocytes following hypoxia ischemia in developing male and female brains.
Dissect the brain from the head of a newborn mouse pup by using small scissors to create a posterior to anterior midline incision through the skin to expose the cranium. Cut the cranium midline carefully from the neck to the nose. Use sterile flat-tip forceps to peel away the skull. Remove the brainstem with the help of curved forceps and place the remaining brain into a dissecting dish. Using curved forceps, flip the brain so that the ventral surface is facing up, and then separate the hemispheres with a sharp, sterile surgical blade. With flat curved forceps, peel back the cerebral hemispheres and carefully remove the bulging midbrain and thalamic tissue to reveal the hippocampus, a small seahorse-shaped structure in the medial temporal lobe. Remove both the hippocampal lobes and carefully clean the meninges and fibroblasts from the surface of the lobes by pulling with fine forceps. Transfer both hippocampal lobes from a single mouse into a second dish filled with approximately